Right, so that was a great pleasure to introduce Aubrey Weigel from the Janelia Research Campus. So Aubrey will be talking about her sort of latest work as the lead project scientist at the on the cellular organelle segmentation in electron microscopy or COSM project at Janelia. But by way of background, it's just worth pointing out that Aubrey really sort of started off her postgraduate work as a single molecule biophysicist, so tracking single molecules on living cells, and what have you, looking for their sort of biophysical properties such as anomalous diffusion and so on. And I think in those days, you know, this was a tricky thing to do, because really you're trying to look at a moving object on top of another moving object, the cell. So this is a very a tricky sort of measurement to get right. So I guess from studying the sort of biophysical properties of single molecules in this, Aubrey moved on as a postdoc into the Lippincott-Schwartz labs, then at NIH, and basically started looking at these sort of biophysical properties in a sort of more cellular environment. From there, with a sort of big leap now, Aubrey is now leading the sort of segmentation of SEM imaging. So quite a broad portfolio of skills that she's brought to the whole problem. I would say yet another big tricky problem, how to deal with EM data and automatically segment it. So I guess without sort of going into too much, it was sort of using machine learning and techniques like that. But I guess I should really just hand over to you, Aubrey, and let you tell us about your work.
Yes, thank you for the introduction. So I'll share this here. Can you see this okay? All right, so yeah, so I will tell you about the work that I've been doing recently in automatically segmenting whole cell organelles in volumetric electron microscopy. So just as a little bit of introduction, because sometimes it's nice to take a step back and look at why we're doing things, right? So if we take a look at the cells, you know, a defining characteristic of all living things is that they're comprised of cells. So we can look at the cell as a fundamental unit of life. And if we focus in on a cell, it gives us details about and understandings of the tissue and the organism that the cell composes. Now, if we look a little closer inside of the cell, the organelles inside are the building blocks. So from highly specialized cells within a tissue to plant cells to immortalized cancer cell lines, even simple unicellular organisms such as protozoa, at the foundational level, all of these organelles within the cells have the same basic functions. So for instance, DNA is packed in the nucleus, proteins are synthesized on the ER, and the mitochondria generate ATP to fuel the cell. Historically, to look at these organelles inside the cell, we've turned to light microscopy, which has brought us a long way in understanding organelles and their functions. The beauty of fluorescence microscopy is it grants us protein specificity. So it allows us to see organelle dynamics and how they interact with each other with global context. So we can see all the organelles that are labeled in the entire cell. However, we are limited by the protein labels and resolution, so there's a sampling problem. EM, on the other hand, has also taught us a lot. It offers global contrast and higher resolution, but traditional methods usually limit the volume to which we can investigate, so we lose that context. So even though we can see everything at super high resolution, it's usually a small volume, so we lose that whole cell context. So through the work of the Hess lab, the Fibsome focused ion beam scanning electron microscope has really brought us a long way, because now we can get near isotropic resolution, we can get global contrast, and we can do this across large samples with the enhancements that the Hess lab has made. So not only do we gain resolution, but we also retain that context. So here's an example of what some of those Fibsome volumes look like. So I have 4 different samples here. I think a HeLa cell up here on the top, a pancreatic beta cell here, a mouse choroid plexus here on the bottom, and then a Drosophila ventral nerve cord here at the top. So you can see that they all have a little bit different flavor, but all of them are packed with organelles and information that's just waiting to be mined. What you might have noticed when we take a closer look is that there's a ton of information. Even in this block that I'm showing here is about 1 cubic micron. You can see that there's almost no free space. There's stuff everywhere inside of the cell. So you see in this cubic micron centered around the centrosome, we have the Golgi, the ER, the nuclear membrane studded with pores, the chromatin, lysosomes, autophagosomes, microtubules, ER, vesicles. Everything is right there, and this is only a cubic micron. So you can imagine that this takes a lot of work to get the information out of the EM data. So having all that information comes at a cost. To put that into perspective, it took a manual annotator or an annotator to manually annotate all of these membranes about 7 to 10 days, and that's kind of a generous estimate as to how long it took. It can easily take up to 2 weeks to do this sort of block. So if we extrapolate that to an entire cell, that would be 50 to 100 person years to manually annotate just one cell for all of these different membranes. Obviously, this is not feasible. So enter COSM. So at Janelia, we have some of the world's leading experts in machine learning, cell biology, and large volume, high resolution EM data acquisition. And at their intersect, it's a really unique area of research. COSM is expanding that intersect and driving forward our tools and our knowledge with respect to identifying the subcellular structures within EM data in an automated way. As a result, the products that COSM provides includes quantitative analytics and the data resources. And it also allows us to reimagine our canonical textbook image of the inside of a cell, provide biological insights and knowledge, and do this in a way that is open and accessible to the broader scientific community and to the public. So what does that look like? Well, we're a highly collaborative team, and it includes members from scientific computing within Janelia, project technical resources, as well as individual group leader labs, such as the Saalfeld and Funky labs. So our pipeline begins after the data has been collected. We start with the raw EM data and pre-process it to align all of the stacks so that we have a nice complete Fibsome volume. We're using software implemented by the Saalfeld lab, along with Stefan Preibisch at Janelia to do this. And from there, skilled annotators then take the data and carefully classify each and every voxel within a small block, similar to that 1 cubic micron that I showed you before. These segmentations are then fed into machine learning algorithms as training data, and the prediction output from these algorithms are then evaluated. So we have to find which network did the best. They're refined. There's always improvements that can be made. And in some scenarios, we use those prediction outputs as a tool for other pipelines, such as CLIM registration. Once we have those predictions, the whole cell segmentations are achieved, and the quantitative analytics of subcellular distributions, morphologies, interactions, sizes, et cetera, can all be acquired. And then lastly, and most importantly, I think all of this data and software is made publicly available for viewing and download on our data portal, openorganelle.org. So I'll walk you through some of the steps of this pipeline using one of the HeLa cells as an example. So we'll start here with the aligned EM data. And from here, Grace and Allison then manually classify each and every voxel in the subblock. We have up to 37 different classes of substructures. And then here is an example, maybe if it goes, there. Here's an example of each of those substructures. So whenever possible, we try to distinguish between the membrane of an organelle and its lumen. So for instance, the mitochondria membrane and the mitochondria lumen. So we include all distinguishable membrane-bound organelles from the popular and easier ones, such as mitochondria in the plasma membrane, to trickier ones like the ER and Golgi lipid droplets and the endosomal system. We also include cytoskeletal elements like microtubules and nuclear components such as chromatin, the nucleolus, and nuclear pores. And we also classify the ribosomes within the data sets. So shown here are 15 of those subblocks where Grace and Allison have manually annotated every single voxel within the block. The class labels are color-coded on the left there. And then all 37 classes are shown in the graph on the right, which depicts the amount of training voxels that we have. And I'll note that it's in log scale. So you can see that there's a really big disparity between the number of voxels between some classes. And then here on the bottom graph, I'm showing the different, where the training data comes from in terms of the data set. So you can see we have 5 different cells that we're acquiring from. And even in our most generous sample, this JRC HeLa 2, it's still less than 1% of the entire cell that's being annotated. And then lastly, I'll point out, you'll see that in this graph of the training voxels, it's color-coded green, gray, and with white dots. So we have 3 different networks that we're using to train. And we ran an experiment to see whether it's better to train all 37 classes at once, which is all of these bars, whether it's best to train only from classes that are often seen together. So for instance, like ER, mitochondria, nucleus, plasma membrane, et cetera. And those are shown in green. And then we also trained individual networks. So just ER, just plasma membrane, just nucleus. And those are shown with the dots here at the top. So we have an all network, a many network, and an individual network that we trained for these. All right, so what do I mean by training? So the training data I just showed to you is then used to train machine learning classifiers. We use a mostly standard setup with a 3D UNet architecture, as you can see here in the middle. And this is trained by repeatedly feeding in blocks of raw data that produces some output for the innermost part of the block, so the orange part here, which is then compared to what it should look like according to the ground truth annotations, and the parameters of the network are adjusted accordingly. So this process is repeated hundreds of thousands of times in our case. Specifically, we train it to predict a sine hyperbolic tangent distance transform of the binary label for each organelle. So we're predicting distances, not a binary label segmentation, which is different from most segmentation approaches. So you may or not remember or know that this was the same approach that was used to predict synapses in the FAST-B data set for the Drosophila brain. So these distance transforms can then be transformed back into that binary segmentation that we're used to by just thresholding at 0. So here's just an illustration for the mitochondria, which is in blue, and the ER in green. But as I mentioned before, we train on the number of these networks where we change how many organelles are actually trained simultaneously. So we have a setup where we train all classes together, one where we only include the classes that are covered by a lot of the training blocks, and then also the setups that are just a few classes at a time. So all mini and individual. So to find which setup does the best and what iteration is optimal for each network, we need to evaluate the output in comparison to the data. We experimented with a couple of ways of doing this. So first is a classic approach of computing the metrics on additional annotations. So for this, annotators manually annotated a 4 cubic micron holdout block in each of the 4 data sets. So this is an example of one of those 4 cubic micron blocks. We then pulled the predictions on that holdout block. So this is the raw predictions from that same region. And then we evaluated on that block. We use the F1 score, also known as the DICE score, which falls between 0 and 1 and is based on measuring pixel overlap. So the higher the score, the better. The results I'm showing here are for a subgroup of the organelles where we have directly comparable data for each network type. So what's important to note here is that for this sort of metric to work, we have to have all the organelles that we're comparing across all the data sets in each of these holdout blocks. Otherwise, we're comparing apples to oranges. So for each network, there are 4 points for each of the 4 data sets, rather than an average over all the 4. So you can see the spread between the 4 data sets. So for instance, here for the plasma membrane, you can see with the all network, the 4 data sets all performed around 0.8. So generally speaking, we saw no large effect, but overall there was a hint of a trend, which we have later verified experimentally, by having more classes together, training on more classes together seemed to help the network performance. Some of the large spread that we see for some of the organelles has 2 possible causes. So for instance, with the mitochondria, the obvious scenario is that there's a real difference in performance. The less obvious, and what we think to be the more likely, based anecdotally by looking through our data, is that the sampling of the space is poor when using validation blocks. So for instance, if you do not have an entire mitochondria in your validation block, or if you only have a small part of a different looking mitochondria, then you're going to perform very poorly for mitochondria within using that validation block, whereas the rest of the cell with all the other organelles might be doing really well. So this is a downside to this approach, because we can only directly compare structures in this manner that are covered by all the validation blocks. Also, there's an overlap based measure such as this, which can appear overly pessimistic for thin membrane structures. So because it compares almost a voxel to voxel metric, you can imagine if it's something really small, like a microtubule, for instance, if you have a slight displacement of the voxels, then it's going to give you a very poor score, when in reality, the microtubule might be completely reconstructed and actually did well. But maybe most importantly, one of the biggest downsides to this is that this is extremely costly. So if you remember, for the training blocks, what we use is a roughly 1 cubic micron block. And for this, to be able to get enough information into that validation block for it to be useful, and even then it was on the almost too small side, we had to do 4 cubic microns. So to do this for any data set that you want to test is really costly to have generated. So we're talking months of work just to get a validation block, which makes it really hard to go into new data sets and just blindly pull predictions and get results. So for these reasons, we turned to another approach, which we seem to favor more. So what we did is we relied on a more manual evaluation by doing pairwise comparisons. So to do this, David and the group implemented a custom VG plugin for us to pairwise compare 2 different predictions at a time. So the user first selects 2 predictions to compare, as well as an organelle class to evaluate. So I'm showing the ER here. The user inspects the stacks and selects which prediction, if any, did a better job of predicting the chosen organelle. So I'll note here that the crops are randomly labeled, so the user is completely blind to which image came from which prediction. And the plugin calculates and reports a p-value for us, which corresponds to the null hypothesis that the 2 networks perform equally well. So this evaluation process then repeats for a new crop, and the evaluations are performed until after a minimum of 100 evaluations, we reject the null hypothesis, and the network that performed best is deemed the winner. So we don't allow any ties, we force a decision to be made. So we use this manual evaluation to back up the conclusion that we reached with the automatic validations. And that is that most of the time, the combined networks, so training on all 37 classes at once, seems to perform better than when we just train on 1 class at a time. So the more information, the better, was the takeaway. Using the setups that optimize via the manual validation, we can then generate the predictions on the entire data set, the exciting stuff. We threshold them and thereby produce the segmentations. And those segmentations aren't quite perfect yet. There's always room for improvement. But some of the errors that were happening were relatively straightforward to address by applying techniques such as size filtering, picking only the largest connected component, which works for like the plasma membrane, for instance. Or we do things such as a watershed algorithm to correct overmerging or oversplitting of organelles such as mitochondria. So pretty simple stuff for cleanup. In some cases, though, we're able to leverage a priori information about the organelle. So for instance, this works really well for microtubules. So Nils from Jan Funke's lab was able to improve the predictions here quite a bit by applying a technique he developed that incorporates structural biases for the microtubules. This method depends on solving a discrete optimization problem with the associated hyperparameters. So first, a data-dependent evidence prior. Second, a stiffness factor that controls how much deviations from a straight line are punished. Third, a start edge prior, which defines the cost for starting and ending a trajectory. Fourth, a negative selection prior, which is needed to avoid the trivial solution of selecting no edges and no vertices. And last, a data threshold for the candidate extraction, and then also a distance threshold below which the two candidates are connected by the candidate edge. So all these hyperparameters are used for each cross-validation run and found through a grid search over 2 out of 4 densely traced 2 by 2 by 2 micron microtubule validation blocks. So that's what's shown in red here is those blocks that were used for validation. And then the result of which is shown, the result of all this reconstruction is shown here in blue. So we wanted to make sure that all of this work was worth it because it was quite a bit of work to train another network and implement, generate the validation blocks, et cetera. So in order to test the applicability, Nils compared his method, which is in blue, to that of a baseline method, which is in gray. So is it worth the extra effort? So the baseline consisted of very similar to what we did for our base refined predictions. So he thresholded the microtubule predictions. He did morphological closing of the thresholded predictions. He then ran connected component analysis and size filtered those connected components. And then he did a skeletonization of each connected component where he restricted the skeleton to have no branches. And then he directly compared that baseline to his reconstruction efforts. And what he found is that in each case, you can see that Nils' method did perform better. So it was worth the effort. And we evaluated this in terms of topological errors, such as the false positives, false negatives. It performed especially better with splits and also with merges. And then we also evaluated using precision recall and F1 metrics. And again, the results were in agreement with the topological error evaluation and that the refinement showed improvement over the baseline refinements. So we used these refined microtubule predictions, which were actually skeletons up to this point. We then artificially expanded that skeleton to create a tube that had an inner radius of 6 nanometers and an outer radius of 12.5 nanometers, which would be consistent with experimental measurements of microtubules. And then we combined all of these predictions and refinements and able to get very nice segmentations for a number of the organelles. So I'm showing you the results of those segmentations here. And this worked pretty well across the 4 data sets that were our main focus for starting out. So we have 2 HeLa cells, a macrophage, and a ChurCat. What's really exciting about these predictions is we can start to see organelles in a way that we've never been able to do before. So, for instance, we'll take a look at my favorite organelle, the Golgi. So if you look at a textbook shape of a Golgi, it's this cute little stack with 5 to 7 neat little cisternae and a clear cis space and trans space. However, when you look at the entirety of the Golgi in a whole mammalian cell, like we have here, what we find is something that's much more complex, and I would argue is much more beautiful. So those 7 cisternae that we typically see in the textbook images, in actuality weave into and ribbon throughout other stacks. And what may be the first cisternae of one stack becomes the last cisternae of another stack, which is really quite amazing to see in 3D and in reality with this data. What you can also see are the vesicles that are centered all around the edges of the Golgi. And as I'm showing here, the ER is so intimately connected with the system that it can be hard at times to distinguish one from the other. So all these complexities are completely lost in textbook cartoons and nearly impossible to resolve from light data alone. These segmentations, however, are more than just a pretty picture. We can now start to quantify that information from them as well. So for instance, we can look at straightforward things like the volume occupied by each organelle class across different cell types. So you can see the nucleus occupies the most space in the cells, maybe not surprising, but this is especially so in gercats, which are an immortalized T cell of sort. So also the ER occupies a much larger volume in the macrophage cell, you can see down here, which agrees with it being a highly secretory cell. So I'll walk you through 3 additional analysis examples now. First, we'll look at the contact sites between a microtubule and other organelles in the cell. So you can see the microtubule here and color-coded are the organelles that it comes in contact with. So this particular microtubule is only about 1 micron in length, and it's really busy. We can quantify these contacts for all of the microtubules in themselves, and the results are down here. So you can see it's in contact mostly with the ER, which is not so surprising. Next, I'll show an analysis of membrane curvature. So for this, David took ER predictions and iteratively thinned them down to produce a medial surface, a skeleton of sorts. He then could calculate the curvature that the surface and the threshold, the curvature, and he thresholded that between planar, which is in green, and tubular, which is in purple, regions of ER. So that's quantified down here for HeLa and macrophage. So anecdotally, we noticed 2 things. One, that the highly secretory cell, like the macrophage, has more planar peripheral ER regions than the HeLa cell. And 2, there seems to be an affinity between planar regions of the ER and the mitochondria contact sites in the HeLa cells. So as you can see when the movie paused here, the contact sites in blue seem to match up to the mitochondria in orange. And when we quantify that over here on the right, what we see is that is actually happening. So the third analysis I'd like to show is an example of the ribosome classification. So we start with all the ribosomes, there's lots, and then David classified as those being cytosolic or ER bound, and then further categorized them as being on planar regions of ER or tubular regions of ER. And what was interesting is that we found that the ribosomes on the more planar regions of the ER tend to form those beautiful polyribosome chains. So you can see some of those here in these flattened regions of the ER. This, interestingly, was corroborated with live cell imaging of ribosome translation and ER markers that's been being done in the JLS lab. Okay, so everything we've shown so far has all been on very high resolution data. So the voxel size is about 4 nanometers isotropic. However, a lot of the data that is coming out of Janelia and on Fibsomes is actually 8 nanometer isotropic data. As you can imagine, it takes a lot less time to acquire a 8 nanometer sample than it does a 4 nanometer sample. So we wanted to make sure that our networks would also work on that data. So what Larissa did was she reused the 4 nanometer training data to train a second set of networks where she roughly simulated what 8 nanometer versions of that raw data would look like by randomly subsampling the data on the input side. So with those networks, we were able to produce 4 nanometer segmentations on 8 nanometer data. So here are the evaluations for the 8 nanometer networks compared to our 4 nanometer networks. So again, we see pretty similar performance between the 2 networks, which is cool. An interesting observation as to which network has the upper edge seems to be aligned with the resolution field of view trade-off. So as you can imagine, for big objects like the nucleus or mitochondria, the more you can see of the organelle, the easier it might be to segment that. So for 8 nanometer data, you'll be able to see more of the nucleus, more of the mitochondria at a point in time, or as it's training. And so it does a little bit better, it seems, when predicting those. However, for something that's very thin, like microtubules or mitochondrial membrane, we start to lose some of that information. And so the 4 nanometer networks seem to outperform in those situations. Some things to note here, though, is that the optimization is on the other 3 holdout blocks that are from both the iteration and the network type, whereas in the 4 nanometer evaluations I showed earlier, it was just for the iteration. This also does not include the all network, so this was just for the mini and few network because we don't have that fully evaluated yet. And the evaluation for the 8 nanometer networks works in the same manner as the training. So In other words, we predict on a randomly subsampled data, and we're aware that this might not reflect real 8-nanometer data, but it also comes at a cost of not having to provide validation blocks for all of these samples. So on the bottom here are the results of this. So we have another HeLa cell, which is at 8 nanometers. So it should be pretty similar to our 4 nanometer data, and the segmentation seemed to work great here. But what was really encouraging was when we looked at something that was completely different. So for instance, we had a mouse choroid plexus appear on the right. And this data set, not only is it tissue, but it was also chemically fixed. So it was prepared in a completely different way. And the networks did a really nice job of predicting the organelles in that as well. So that was encouraging for us. All right, so another interesting aspect is that the 8-nanometer networks allowed us to get segmentations for existing correlative light and electron microscopy data sets. So here, John was able to use them to automate the alignment of the EM data to the light microscopy, as you can see here for a COS7 cell. So we're zooming in here, and what you'll notice is that there's the deformable part of the alignment as we're switching between the original Fibsim and the Fibsim aligned to the light data. So more specifically, John used the mitomembrane segmentation, which will show up in orange in a second, to generate images that look like the LM data by downsampling and applying an anisotropic Gaussian blur to mimic the point spread function of the light microscope. So these images are then automatically aligned to the light microscopy mitos, which were in blue. And as you can see, they match up quite nicely. All right, so let's look a little more closely, and I'll slow down and explain that again, because I know I ran through that quickly. All right, so John starts with the fluorescent mitochondria membrane data, which is in blue. And then he uses the predictions of the mitomembrane in orange here. And he blurs the mito predictions in orange to match that of the blue. So he takes the isotropic Fibsim predictions and he blurs them more in Z than he does in XY to match the point spread function that would come from the light data. He then registers the two. So that would be the same as registering the EM to the LM. And as a result, then we can look at the ER, so the light data of the ER and the Fibsim predictions of the ER. And this would be an independent output of that alignment. So if the ER is in good agreement, then that means that we did a really good job because that's independent of what we're using. All right, so we looked at the Jacobian determinant of the transformation field to measure the local distortion resulting from registration. So as you can see in both a single slice and the histogram over the whole volume, the distribution has a nice and narrowed standard deviation, which indicates a smooth transformation field. We also evaluated the registration accuracy with respect to human-generated ground truth. For this, we used the ER to compare, since this is not used for the automatic registration. It's completely independent. So this enabled us to measure errors in an unbiased way with respect to the true underlying transformation, not only the part of the transformation that can be inferred from where the mitochondria membrane channel lies. So the way that we did this was kind of fun. So John manually placed 31 landmark pairs and aligned them to the ER and the LM and EM data in this box region. So wherever you see a white spot, John picked a point, and then he aligned the two channels, the mitochondria EM and the ER, EM, and an LM. He then gave me those unregistered images and half of the pair. So I only knew where the landmarks were, and I had to align them myself. And then I aligned the EM and the LM data. So after I aligned the ER in the two images using his landmarks, we compared our results between the two of us, as well as our results to the automatic registration. All right, so you can see here that the horizontal is how well our registration compares to the automated registration. And then the vertical is how well we did compared to each other. So you can imagine, since remember, we're aligning the ER in places where we have really clear ER signal, John and I probably did a really good job. And that's true. So you can see in the upper right here, there's a short line for the human versus human registration. So there's nice, clear ER signal. It's very easy to tell where we would need to align the images together. However, over here in the bottom, you can see where the ER gets, where there's not as clearly defined ER structures to align. John and I had more disagreement with each other because it's more ambiguous as to what we were going to be aligning. And then, but conversely, I guess, or mirror to that, if we look at how we did comparison to the automated registration. So here we're comparing the mitochondria signal, right? Because the automated registration is using mitochondria to align. So in places where there is clear mitochondria signal, the automated versus human alignment did very well. So again, if you look down here where there's a nice mitochondria signal, there's a very small error between the automated and human. However, if you look back up here where there's no mitochondria to be found, the automated versus human accuracy is much less. So all in all, the difference between the human annotator and the automated registration was on average about 110 nanometers. So I'll note here that while that may sound like a lot, that's actually only 2 to 3 pixels in the light data. So that's actually not very far at all. And what I would like to point out though, is that John and I differed from each other by an average of 30 nanometers. And while this really isn't that much, it also means that we can't expect to get much better than that if 2 humans can't even agree more than 30 nanometers or more closely than 30 nanometers. All right, and then lastly, we compared the independent registrations of the EM to both the PALM and SIM. So you can see here a map of the spatial error between the 2 transformation and the histogram of spatial error over a region where cells are present, which we outlined in white. So overall, the errors are small, especially near the mitochondria, which is to be expected, which indicated to us that the registration is consistent across the modalities. So altogether, we showed that using the automatic organelle segmentations can be used for successful registration of CLIM images, which make it accessible to less experienced users and reducing the time from days to an hour or less, which is substantial. I should mention though that this is the best case scenario. The extent to which the alignment process can be automated really depends on the quality of the data, always, right? So sometimes we'll have to manually tweak things such as masking out a cell that is not transfected in the light, or in the worst case scenario, just doing a manual alignment task, but using the segmentations as a guide. And I can say, having personally had to align several of these data sets with and without segmentations, that even having those segmentations is a huge help for this. All right, so now I want to take just a moment to give you a little bit of a vignette on how this data has been used to answer some biological questions. So this work is what I did while I was finishing my postdoc in the lab of Jennifer Lippincott-Schwartz together with Qilin Chang. We're interested in the structures that are called ER exit sites, the site where freshly synthesized proteins exit the ER and are then transported to the Golgi for further processing before reaching their final destination. To do this, we used a cryo-cleanse system similar to what I just described for the automatic registration in the last slide. So quickly, what happens here is we have a sample that is high pressure frozen. And then while still vitreous is imaged under cryofluorescence. So in this data sets that I'll be showing you here, we use structured illumination microscopy. And then that frozen cell is then freeze substituted, resin embedded, and prepared for Fibsome, and then directly imaged with Fibsome. And then we have those 2 correlative data sets to compare. All right, so to first identify the exit sites in this data, we used a COP2 protein, SEC23, and imaged it under cryo-sim. So this is a U2S cell that's overexpressing this COP2 protein, which will pop up here in blue and green. And you can see as we scroll up and down, we get the canonical exit site morphology that we'd expect from TEM images, specifically these kind of bubbly vesicle-like structures in the EM. All right, but the beauty of the Fibsome now is that we have isotopic volume information as compared to what we did with TEM. So we can start to look at these exit sites with 4 nanometer X, Y, and Z resolution. So you can see that all the black dots here are the ribosomes and actually make the ER easy to identify. And they help make an arbitrary distinction between the ER exit site in the ER because the exit site membrane is always void of ribosomes. So interestingly, what we find is that the exit site is continuous with the ER as a single globular structure. So it's like this hand of tubules all intertangled, reaching out of the ER, which is in contrast to what we've seen in traditional electron microscopy images. So furthermore, we use light microscopy to quickly verify that the ER is continuous with the exit sites by using fraps. So we frapped a cargo that was caught in the exit site, and we saw that it quickly recovered back into the exit site. So it corroborated the information that we saw from the EM that they are continuous with one another. We were interested in what happens when we overwhelm these structures. So to do this, we used a rush system developed by the Perez and Boncompin labs. And so quickly what happens here is we have an ER hook and the cargo, which is connected to that hook through a streptavidin binding protein. So it's caught in the ER, you can see it all over the ER here, and then localized in exit sites. And then if we add biotin, it will outcompete that streptavidin binding protein and release all the cargo at once in a rush. And so when that happens, you can see all of the cargo rushing out of the cell. So what we did is we expressed this in our cells, and then we did cryoclam 8 minutes after release so that we'd get a cell that looks similar to this. So this is the result of that. And I'll skip ahead here for the sake of time and zoom out to the periphery. So we're going to go out to the periphery of the cell and look at one of these specific ER exit sites. There, okay. So if we zoom in, you can see that, again, as in all the other data sets that I've shown, you can see that it's full of membranes and ribosomes and cytoskeletal structures. And you may notice in the center that those little bubbles that look like the ER exit site popped up. If not, we can turn on the rush cargo, which will fill up that exit site. And you can see it right here. And if it's still hard to see, we've also turned on the segmentation here, which will come up in the next pass through. So you can see the exit sites connected to the ER, which is kind of moving all around in that area. The exit site itself is segmented in green now. And when we do the segmentation, what we find is that there are these kind of tubular structures all connected and then connecting back to the ER itself. So what was nice is with this sort of information, we can now directly compare cells that have this overexpression of rush cargo with cells that are untransfected. So we did this comparison, and what we saw anecdotally is that the overwhelming the cell with this rush cargo seemed to swell the exit sites. And then when we measured this across all the different cells, we saw that this was true. What was surprising here was that not only did it grow the exit site itself, but also the tubules that were within the exit site. So as the cargo built up in these exit sites, we saw that it almost doubled in size with the tubules themselves and the entire exit site diameter. And we measured this across several different cell types as well. All right, so the next really interesting thing that we were after is what happens when that cargo is then released. So if we turn back to a live cell microscopy image, 8 minutes after release, remember there are all these tubules reaching out of the exit sites that are reaching towards the Golgi. So these are all transport intermediates. These are the carriers that take the cargo from the exit site to the Golgi. So we turned back to our cryoclam system and looked at one of these carriers in this system. And so we'll go here now to the top of the cell. What's nice about this region is it's nice and flat. So I'm looking right above the nucleus and the plasma membrane in this really thin section. So it's nice and parallel, which is convenient for viewing purposes. All right, so as this scrolls through, we're starting at the top. So we'll go through the plasma membrane. You can see all the cortical actin there. You can see some of the ER membranes, the polyribosomes on the nuclear envelope, the pores, and now we're down into the nucleus. You can see the chromatin. You might have noticed on the right that there is an exit site. right there, highlighted in green. And if we turn on our cargo fluorescence, you can see that the cargo fills up that exit site. And emanating out of that exit site is this pearled membrane, which is really quite peculiar. And so this pearled membrane is actually the transport intermediate. So we could see the cargo in the exit site, that pearled membrane reaching out of there. And then excitingly, this is all along a microtubule as well. So you can imagine if this were in a live cell, this would be this transport intermediate is being pulled out of the exit site along a microtubule on its way to the Golgi. And so because we have the wild type data, we can also look at this in, well, I guess I should mention that there's plenty of these across an entire cell. So here are 10 such transport intermediates within just this one quadrant of the cell that we identified, all having this pearled membrane feature. And then we are also able to see this in an untransfected cell. So this wasn't just the result of over-transfecting this rush cargo. So I'll skip ahead here so you can see the main result. So you can see this huge pearled membrane being pulled out of the exit sites and always along these microtubule tracts, which is really spectacular. All right, so from this little vignette, what we're able to see is that the nanoanatomy of the early secretory compartments in intact mammalian cells, that the exit sites were these tangled tubule networks that were connected to and exchanging with content from the ER. The sequential action of COP1 and COP2 happening at the exit sites for the movement of cargo, and these pearled membrane vessels moving along microtubules for Golgi delivery. Okay, so maybe the most important part of all this work is that we work really hard to make this all publicly accessible and viewable. So including the work that I just showed of the exit sites, all of this is available on our data portal, Open Organelle. Here together with the HES Lab and Fibsome Technology Group, we provide browsable and very soon to be queryable, hopefully later this week, interface for people to explore these data sets and the accompanying segmentations. So you can browse pre-made views or customize your own views based on what you want to see. And towards the end of this week, we should also have 3D visualization. So you'll be able to see the 3D rendering of the segmentations provided as well. So it's all made possible through NeuralGlancer visualization. This data is set up in a multi-resolution pyramid schema, which allows for fast online browsing as well. And we provide tools for anyone to download any of the data or view crops of the data, however they like to, including the raw data, training data, segmentations, and the accompanying analysis. And the SOFA lab has also implemented In5 capabilities in Fiji. So now in the base Fiji installer, there is an In5 reader. And so you can directly read from S3, and that information is actually available here. It'll show the dataset location. You can click on the Fiji button. It'll copy that location and you can read that from your Fiji browser directly on your desktop. So we have tutorials and provided a glossary of sorts as well, containing different examples of each of the organelles that we've classified. So you can see how we defined each of the organelles. I mean, we link all of our GitHub repositories, and all the information on the tools, the models, and the source code is available there as well. So of course, the ultimate goal of COSIN is to segment organelles and EM data, but there's a lot of work to be done here. This is only just the beginning, which is the exciting part. So we now have a pipeline in place for getting us started. And while we of course try to continuously improve every aspect of that, we're also seeking to understand what else is needed to maximize the impact of this work. So for instance, how much training data is really needed for a new data set? How much proofreading is required? How generalizable are our analysis methods in new data sets? And can we leverage light microscopy for training and or validation? I'm showing you just a few of the collaborations we're working with, including some in Janelia, some with the Advanced Imaging Center, and then also with Genentech to explore these areas. So all this work was made possible by an incredible collaborative team effort and can be found in our preprint on bioRxiv. And really, this would not be possible without the larger support of administration, lab support teams here at Janelia, both past and current. I think missing from this list is Amalia, who has done a lot of work, and I think is on this call, and really the foundational sample prep, which you see the images wouldn't be really here without her work. But yeah, this all takes an army, and this is the result of those efforts. Thanks.
All right, thank you very much. That was a fantastic talk. I guess, do you want to do the Mentimeter now, Stephanie?
I think we are probably having more questions than normal, so I think we should just go with the questions actually. Sorry, I was a bit late today. I'm
Okay.
Stephanie here, so I prepared actually the Mentimeter, but I think maybe we have only 10 minutes, 9 minutes. Should we just kind of,
Yep.
There were so many questions.
I'll go into the questions. Actually, just quickly before I launch that, I just want to point out next week is a bank holiday in the UK, so we won't be here. Following that, though, on the 10th of May, Gail McConnell will be talking about using the mesolens for optical mesoscopic imaging with subcellular resolution. So that's in 2 weeks' time. Thanks. Okay, Aubrey, what I'll do is I'll just read out some of these. Paul Vacado, are you there? Do you want to say your, ask a question yourself?
Yeah, that's fine, I'm here. Hi, Aubrey. Excellent presentation, lovely work. I had a question about, you mentioned about the microtubules, that they interacted with organelles a lot of time. Is that really interaction? Do you know, especially in the first example? With
Yeah.
I'm convinced, but.
Yeah, so the way that we defined that interaction was based on spatial distribution, and we chose a distance that was the length of typical microtubule motors. So, of course, there could be an organelle that is, that distance away from the microtubule, and that would be a false positive in this situation. But we tried to keep it within a small range of them touching each other to make those claims. So yeah, really, this is kind of the gateway drug to speak, right? So you can get this information, see cool possible interactions, and then go back to your classical light microscopy or choose your favorite microscope setting to really test these and corroborate them, right?
Okay. One quick follow-up on this, which is about your technology. I'm assuming your high pressure freezing is essential. You've never seen, so to say, the tubulated ER in chemically fixed samples. Have you checked?
Yeah, so we see these structures. I mean, we showed this previously in prior work that I had done, but you can see this in both chemically fixed and high pressure frozen samples. So it's not an artifact of either. I think the high pressure frozen, well, without chemical fixation, you do get some better preservations of membranes. They both have their pros and cons, but it really depends on what you're looking for. But I think with all of the variously fixed samples, and most of them that I've shown here were high pressure frozen, but by all means, that's not the only thing that we use at Janelia, not the only thing that we've segmented, but these are pretty standard throughout.
All right, thanks very much. So next one was from Corny Klevegt. Sorry if I've messed up your name. Are you there? Do you want to ask?
Yes.
Oh, here we go.
Thanks, Aubrey, for the presentation. I was wondering, so what specific machine learning algorithm did you use? Was it TensorFlow, and what implementation for the convolution part of the network did you use to gather together the 3D information into probably a 2D machine learning algorithm?
No, everything was three-dimensional. So it was maintained at 3D the entire time. This is quite obviously my weakest link, and really where I relied on my collaborators' expertise. So you can find all this information. If you can't find it in the preprint, then it would also be located in the synapse predictions from Larissa Heinrich and Stefan Schauffeld, but definitely on their GitHub repositories as well.
All right, great.
Okay, great, thanks.
Thank you. And then Ed Rea. Are you there, Ed? Okay, so Ed was asking how many human hours of work went into the training data sets. So if a group wanted to reproduce this technique on their samples, you know, maybe with some different or divergent organelle structures, how much work would you expect to be needed on their data?
Yeah, so this is a tricky question, right? Because what we found is that we're not even close to approaching having enough training data. We're on the very low end of even having enough to get by. What's optimistic is that we've seen that you don't need very much. And once you have, so we try to make it a point to acquire training data from multiple data sets. And what we see is when we go into a new data set, we only need like 1 to 2 cubic microns worth of data to really drastically improve the performance. And furthermore, you almost need no new training data if the data set looks very similar. And I'm not saying like a HeLa cell to HeLa cell, more so what the staining and the imaging contrast looks like. So for instance, if the mitochondria are really dark in one sample and not as dark in another sample, then those aren't going to perform equally well. You'll need more training data. How much time? That really depends on, so for us, up to date, we've been using small regions where every voxel is classified. This is very costly and very time consuming, but it's what was needed to get all of these classes established so that we could train them all at once. So like I said, that cubic micron I showed, which is probably one of the most complex regions, can take up to 2 weeks for one person to annotate. Less complex regions, so you can imagine out in the periphery where there's not as much going on or close to the cell membrane, those could take a few days. So we had 2 annotators at any given time, plus myself, that was doing all of these manual annotations. It was a lot of work. But now, moving forward, once you have your foundation established, right? Like we have our networks trained, we have those base training blocks, we can now move into a regime where we use the proofreading or the full cell predictions as to bootstrap our training. And so really, I would almost suggest adventuring in that realm if you can, to use predictions that are already there and correct them rather than having to start from ground zero, if that makes sense.
Yep. Okay, thank you very much. And then Yusha said, is there access to this tool and software that we can use for our own data? But I think you answered that one at the end of the talk. So Hanneke says, are you there, Hanneke? Do you want to answer your question? Okay, so would it be possible to use your training data to segment our own Fibsome data? And if so, how will we access it? I think this is sort of what you were just...
Yeah, so definitely you could. The only caveat is that our Fibsome is isotropic, so we don't currently handle anisotropic EM data.
Okay. Well, actually, what would be the penalty there then?
It's not really that it's a penalty. It's just that the way the algorithms are currently written, okay, they have to be isotropic.
Right. Okay, and then User Revik, do you want to ask your question? Are you still there? Okay. So User was asking, what is the tool used in manual segmentation?
Yeah, so for most of the manual segmentation, we use 3 flavors of software. One is Amira, because even though it's pay per use, which we very much dislike, it does have all of the bells and whistles that make these types of annotations nice. We're now moving into using Paintera for most of our segmentations. And then we also use Nosos for the skeletal elements, so for microtubules, for instance.
Right, okay. And then what have we got?
There are so many today.
I'm still scrolling. Benjamin Bacchus wants to ask.
Yes.
Oh, there you go. Fire away, Benjamin.
Thank you very much for the talk. So I was asking about the parameters of the 3D units, but if that's not your cup of tea, maybe I'll just look at the GitHub.
Yeah, what I can say is that so far we haven't been able to fine tune them. So this really is just like the first pass, so to speak, of seeing what we can do with already in place networks. Some of the fine tuning that we did do was set up the experiments of training everything at once versus individual classes. And then we played around with some augmentations, but really haven't gone beyond that much yet. So our first ambitions were just to get this all out there, and now we can start playing with making it better.
Okay, thank you very much.
Okay, actually, I want to ask you a question. So you have 37 different classes, which actually seems to be a pretty astronomically large number of classes to go for. So yeah, so I'm reeling from that revelation. But I mean, is there any scope to make that number bigger? Are there other reliably identifiable things?
Yeah, so the thing is we tried to, so like I said, we're trying to establish, is more information better? And what we found is that yes, it is. So having context helps the networks learn. So really, I mean, I say 37, but you also have to realize that a lot of these are just one organelle. So look at the ER, for instance. You have the ER membrane, you have the ER lumen, you have the ER exit site membrane, the ER exit site lumen, you have the nuclear envelope membrane, the nuclear envelope lumen, nuclear pore inside and outside. You can imagine like everything that's encompassed in the ER, there's actually almost, I think, like 8 to 10 classes that are grouped inside that. So there is a hierarchy that is happening here. Otherwise, we tried to identify everything we could with a caveat of actin. We weren't quite sure how to can reliably segment the actin because it was right in between being able to segment the filaments very nicely to just being too noisy to be able to do something that was useful. So for this first pass, we just avoided it. But we will venture there later. And then there's things such as the lysosomes and autophagosomes and endosomes that kind of get all grouped together because we can't distinguish them by EM alone.
Right, yeah, so it's a sort of an opinion of the annotator in some respect. Although the cytoskeleton was showing up on the periphery of the cell, wasn't it?
Mm-hmm. Yeah, so you can see it out in the periphery pretty well where you have the nice filaments. When you get closer to the nucleus in that more three-dimensional part of the cell, it gets a little noisy, and it really depended on the data set as well. At the time, it was kind of an executive decision that we had our hands full enough with the other 37 classes that we decided to pool our resources there.
Right. Let's see if there are any other questions in this list. I think I've been through most of them. Can I just, okay, so I might have missed this then. So in terms of your sort of correlative registration, do you think 30 nanometers was that the best you did, or did I misunderstand what you were?
Yeah, so the 30 nanometers was how well 2 human annotators could do compared to each other. So within this region, which was arguably a pretty easy region because it was pretty flat and everything was nicely defined, like you could see in the light channel where everything was. So that was how well John and I did compared to each other. For the human versus automated comparison, it was 110 nanometers, which is around 2 to 3 pixels in XY in the light and only 1 pixel in Z. So really we're talking about being off by 1 or 2 pixels. However, with all of these, you can always go back in. So this is all within the big data viewer, big warp environment. So even after you have this automated registration, you could go in and manually fine tune places to make it more accurate.
Right, okay, thank you. I think I just sort of throw it. Does anybody else have any more questions they would like to ask? If you feel free to unmute if you want to.
If you do want a Mentimeter, I need around 25 people to sign in now. Otherwise, we have to kind of soon close this call because we're already 5 minutes over time. So
yeah, I think we might have to just close it. Okay.
It's fine.
Well, I think.
Oh, it was fantastic. I feel like it was just absolutely amazing.
An awkward Zoom round of applause again.
Thank you very much, Aubrey. It was fantastic. It was absolutely breathtaking, totally amazing. Thank you. I mean, everybody, I don't know, you can show all your little faces and say hello, thank you very much, because this is always the most awkward thing about this talk. And yeah, it will be published, the talk, on the YouTube RMS channel, so people can see it. And thank you very much for allowing us to record it as well. But I mean, what a kind of amazing thing to do.
All right.
Thank you so much.
Wonderful. Thank you, Aubrey. Thank you very much.
Thanks, Aubrey.
Thank you.
I think we just want to continue it somehow. Okay. But I think people are leaving now, yeah. So thank you very much.
All right. Thank you all. Cheers.
Thanks.